All right, let's dive right in. Today, we're talking about a really clever scientific breakthrough. This is a discovery that could basically supercharge future medicines, things like vaccines and other therapies, and make them way more powerful. And it all comes down to this tiny, often overlooked part of an mRNA molecule.
So this really gets to the core question that scientists have been wrestling with, right? We all know mRNA tech is a game changer, but the big question is, how do we make it even better? How do we squeeze more power, more bang for our buck out of every single molecule? And that's exactly the challenge we're going to unpack today.
Okay, so here's how we're going to break it down. First, we'll look at mRNA's Achilles heel, a key weakness. Then we'll see the big problem that causes in the lab. After that, it's all about the fix, a really smart segmented solution. We'll check out the test results, see which design came out on top, and finally talk about what this means for the future of medicine. Let's go. All right, first up, mRNA's Achilles heel. So let's start with a quick refresher. The easiest way to think about an mRNA molecule is like a blueprint. It's a set of instructions that tells your cells exactly how to build a specific protein. And at the very end of this blueprint, there's this really important piece called the PolyA tail.
So, what is a PolyA tail? Well, it's pretty much just what it sounds like, a long string of one molecule, adenine, which we just call A. The best way to picture it is like a countdown timer, or maybe the battery life on your phone. Its whole job is to protect that mRNA blueprint and basically decide how long it gets to hang around in the cell doing its job.
And the thinking here seems super straightforward, right? A longer tail means a longer countdown, which means the mRNA survives longer and your cells can pump out more protein. So, simple. To make better mRNA medicine, you just need to stick a really, really long tail on it. But yeah, it's not that easy. In fact, that's where the real trouble starts. And that brings us right to the heart of the issue, the unstable tail problem. Seriously, this has been a huge headache for scientists, a major hurdle that's really been holding back just how powerful these mRNA medicines can be. Okay, just look at this graph. It lays out the problem perfectly. When scientists try to make the DNA templates for mRNA that have a nice long tail, we're talking like 130 A's long, the production process itself just chews them up. See that first bar on the top left? It's showing that for a standard process, over 70% of the DNA templates get their tails shortened. They're damaged. So the very thing you need to make the medicine more powerful breaks during manufacturing. It's a huge problem.
And this isn't some small, random fluke. The researchers spell it out in the paper. They say that these long, repetitive stretches of A's are just naturally unstable. They tend to break apart and get shorter inside the E. coli bacteria that are used to mass produce them. It's a fundamental roadblock baked right into the process.
So what do you do if you have this long chain that just keeps snapping? How do you fix it? Well, that's where this story gets really cool. The researchers came up with an incredibly elegant solution. They basically asked, what if we don't use one long fragile chain? What if we reinforce it? And this picture shows their idea perfectly. See that top design, R1? That's the old way. One long, continuous string of A's. We already know that's unstable. But now, look down at one of the new designs, like S8. See what they did? They inserted these little spacers, different molecules, to break up the monotony. It turns one long, fragile chain into a bunch of smaller, stronger, and way more stable segments. It's so clever. So here's the game plan they followed. Step 1, design and build the DNA with these new segmented tails. Step 2, run it through that tricky production process in bacteria to see if it holds up. Step 3, once they had stable DNA, they used it to make the actual mRNA. And then finally, the moment of truth. Test these new mRNA designs in cells and eventually in mice. Okay, so now for the big question. Did it actually work? Let's get to the results and see how these newfangled segmented tails stacked up against the old standard. Honestly, the results were just incredible. Take a look at this chart. This is measuring how much protein got made in human lung cells. That first bar on the left, R1, that's our baseline, the standard tail. The next one, R5, was an already improved version that gives you about double the protein. But then, look all the way to the right. The new segmented design, S14, gave them a 6-fold improvement, just massive. Let's just pause on that for a second. 6 times more protein. We're not talking about a tiny little tweak here. This is a giant leap forward in how efficient this technology can be, and it all came from just redesigning that little tail. Okay, But as any scientist will tell you, getting great results in a lab dish is one thing. The real test, you gotta see if it works in a living system. So that brings us to the next step, seeing if this champion tail could really perform.
And wow, this image really tells the whole story. What you're seeing are two mice that were given mRNA designed to make a glowing protein. The mouse on the left got mRNA with the standard old school tail. You can kind of see a faint glow, right? But the mouse on the right, that one got the new and improved segmented tail. The difference is, well, it's literally night and day. That brilliant glow is undeniable proof that it's making so much more protein.
So let's just put it all side by side. The standard tail, it's unstable when you try to make it long. But the new segmented tail, rock solid, even at over 200 units. In terms of protein output, the standard tail is our baseline. The new one, it can crank out 6 to 8 times more. I mean, it's just a knockout victory across the board.
Which leads us to the most important part. What does all this mean? Smarter mRNA, better medicine. Why should we care about these incredible lab results? What's the real-world impact? So here's what you really need to remember. First, this new design makes the DNA way more stable, which is a huge deal for manufacturing. Second, it leads to a massive boost in protein production. Third, it opens the door to creating even longer, more customized tail designs. And all of that together means we now have a powerful new tool to optimize pretty much any mRNA medicine you can think of. And let's be super clear, this isn't just some interesting science experiment. Making mRNA more potent could mean we need lower doses for things like vaccines, maybe leading to fewer side effects. It could make a cancer therapy more effective at killing tumors, or even let us go after genetic diseases that we just couldn't touch before. It makes the whole mRNA platform that much more powerful.
So I want to leave you with this thought. For the very first time, we have a reliable way to engineer and build what is essentially supercharged mRNA. Now that we can fine-tune these molecular blueprints with this level of precision, you just have to ask yourself, what's the next disease we're going to tackle?